In Objective 3, we're going to look at the relationship between cell division, cell differentiation, and cancer. Since we will breach the topic of cancer, we're going to define benign versus malignant, as well as the formation of cancer and the progression of cancer. Cancer develops in stages. We start off with an abnormal cell, a single cell that is abnormal, and that cell is going to produce a primary tumor, which will then have the potential to spread throughout the body, forming secondary tumors. Tumors are called neoplasms, and it's basically in a very enlarged mass of cells, and these cells exhibit uncontrolled cell growth and division. So if you have a single cell that mutates and becomes a cancer cell, it's going to start dividing uncontrollably. And that uncontrolled cell growth and division is going to produce a primary tumor. When we classify tumors, we can classify them as being either benign or malignant. Benign tumors tend to be contained. They tend to be held within a capsule so that they can't spread, or contained within a tissue. And these tumor types tend to be non-life-threatening. In fact, the only time that they are life-threatening is when they become so large that they end up suffocating other tissues. If you consider a benign tumor within the brain, how could that be problematic? Well, we know the brain is enclosed in a very tight space within the cranium, within the skull. And if a tumor were to form and start growing there very quickly, you would find that the other cells would run out of space. They would be suffocated. The other type of tumor formation is the malignant tumor, and these tend to be the worst of the two types. Malignant tumors spread into surrounding tissues. We call this invasion, and they start the process of new tumors. They metastasize. This diagram kind of follows the formation of tumors. And again, we start with a single abnormal cell. This is an epithelial tissue, and one cell has mutated, and it starts to divide very rapidly, uncontrollably, and eventually you get the formation of a primary tumor. Primary tumors are going to attract blood vessels. And here we actually see the blood vessels that are growing into that primary tumor. And let me ask this question, why? Why would a tumor attract the blood vessel? Well, to understand that question, you really have to go back to Objective 2 when we talked about cell division. What is required for cell division? Well, the answer is a tremendous amount of energy, a tremendous amount of ATP. So anytime you have a neoplasm, a primary tumor formation, you're going to have the attraction of blood vessels, because blood vessels serve as a highway that is going to bring the nutrients for ATP into that area. So this actually makes the process a lot worse, because you're able to grow a lot faster and uncontrollably since you have this newfound source of energy. When a primary tumor, when it invades and penetrates, it can easily find its way back into that blood supply, back into the blood vessels. If not the blood supply, it can find itself in the lymphatic system, a system that will be covered in AMP2. It's another system that serves as a highway throughout the body. And through that penetration, these cells can travel through the bloodstream, escape somewhere else in the body, and form secondary tumors. All right, this process called metastasis is AKA when cancer has spread, and we all know that once cancer has spread, it's a very bad thing. one of the symptoms of late-stage cancer is wasting syndrome, where the individual loses a tremendous amount of weight, they're very weak, and again, one of the reasons why this occurs is because these tumors are so voracious in the amount of ATP required to sustain their cell division that they literally steal the nutrients from the rest of the body. So late-stage cancer patients tend to be very thin, very weak. They tend to be just wasting away. The other big topic for this objective is differentiation. And this is a difficult concept, one in which we'll spend the rest of the next year touching on. But in order to at least get you started on differentiation, we have to go back to where we all started. And we all started out as a single cell called a zygote. When your father's sperm found your mother's ovum, those two cells fused and they formed the zygote, and this is where you started. And we know that that cell is going to divide into two cells, and those two cells are going to start dividing, and in fact you will divide until you get about 75 trillion cells in an adult body. And during this process of cleavage, cells are also specializing. They're also differentiating. The zygote is going to have a full set of DNA. In fact, they're going to have 46 chromosomes within the nucleus of that cell. And in fact, all 75 trillion cells, all of your cells also have the same 46 chromosomes, a complete complement of the genetic material. The difference between, the different cell types found within the body all depends on which genes are turned on and which genes are turned off. So what are some of these cell types? Well, we know that the adult human has about 200 different tissue types, of which there are fat cells and cartilage cells and bone cells and skin cells and brain cells and muscle cells, 200 different types. These cells differentiate or specialize to become the liver cells, the fat cells, the nerve cells, the muscle cells. So the process of specialization or differentiation is basically when certain genes are turned off, certain genes are turned on, and you end up at a terminal tissue type such as fat, liver, or neurons. So differentiation depends on which genes are active and which are inactive, which genes are turned on and which genes are turned off. We'll pick up that topic later, but for now I think that's a pretty good start.